All right, everyone, welcome to the last lecture for Unit 3. Today we're going to talk about cellular respiration. Now, usually when we have a last lecture, that means we'll have a test coming up soon. Your test looks like it's going to be in about 10 or 11 days or so. Just keep an eye out on Canvas when that is. Take a look at the calendar and just make sure that you're being prepared for all of the material. A lot of the stuff that we cover here in Unit 3 is going to very challenging and there's a lot of steps involved. So just make sure that you're reviewing the material often and taking an opportunity to take a look at it more than once, especially several times before the test is my recommendation. There'll be a couple of activities that we're going to do before the test, and there will be one additional lab that we're going to do. So just keep an eye out for that as well. But we're going to keep on going and just talk about cellular respiration today. Now, last time we had class, we talked about photosynthesis, and basically, this is a really big oversimplification of what we talked about. If you watch the video, you know that it's a way more complicated than just this. This is basically what you learn in regular biology, that when you combine carbon dioxide and water and with solar energy, you get glucose and oxygen. And this is kind of like part of something that we talked about in regular bio called the carbon cycle. You've probably heard of that term before. So the carbon cycle has to be cyclical, so there has to be a second part to it in which the green arrow is actually pointing the opposite way. And so that's what we're going to talk about today. We're not going to talk about photosynthesis. We're mostly going to do cellular respiration today, going from the opposite end. So this is something that animals do. Plants also do this, but mostly animals do this. We do a lot of cellular respiration. We take glucose and oxygen and we get chemical energy from that, and then the byproducts of that are going to be water and carbon dioxide. So that's going to be the big focus today on like how do we go from this to this. And again, this is a really big oversimplification. So we're going to talk about the individual steps. Now, there is an organelle that is responsible for all of this, and that, of course, is the mitochondria. I saw this quote one time, and I thought it was really funny. The day mitochondria went from being the powerhouse of the cell to the ATP synthesis by oxidative phosphorylation was a horrible day. And so that's kind of like a foreshadowing of what the notes are going to be like. I will say that the notes today are going to be pretty challenging, so I would encourage you to watch this video several times. It's really important to understand all the steps involved in cellular respiration and where all the carbon molecules end up going and how we get energy, basically. And so let's just take a look at the mitochondria for a little bit. We know that the mitochondria, a couple of things about it. It's got a double membrane, right? So you can see over here, it's got like a membrane on the outside, like the outer membrane, and then the inner membrane has a lot of folds. And the reason why it has a lot of folds is to basically increase the amount of surface area. Because if you can increase the amount of surface area, then you have more reactions taking place. And that's really good if you want to make a lot of energy. So that's basically what the mitochondria does. Those folds that we see here are called cristae, and those cristae, again, just basically increase the surface area. And then the inner part, this blue part, is called the mitochondrial matrix. And then the space between the two membranes is called the inner membrane space. Okay, so those are the two major things that we have to kind of remember when we talk about the anatomy of the mitochondria. I know there's a lot more stuff with the chloroplast, but with the mitochondria, it's a little bit simpler. We have the matrix, which is the inside, and the inner membrane space, which is just between the two membranes. Okay, so cellular respiration, just a couple of notes. Basically, cells harvest chemical energy stored in organic molecules and use it to generate ATP. Okay, so basically we take organic molecules plus oxygen and we convert that into carbon dioxide plus water plus energy. Okay, that organic molecule most of the time will be a sugar. For us, that sugar is going to be glucose. There are other organisms that can use other organic compounds to basically do cellular respiration. And when we, for instance, ingest starch, like if you ate potatoes or noodles or something like that, starch is the major source of fuel for animals, and then we can break down that starch using hydrolysis. We can break that down into its glucose components and then use that glucose to undergo cellular respiration. So that's the first starting step, is like we need glucose, right? We also need oxygen to start this off. So we're going to take a look at this whole process. But basically, we want to remember that glucose is the starting point. And the path of the electrons in the energy harvest basically means that we are harvesting the electrons from the glucose, from the bonds within the glucose, and basically with the electrons are following this downhill exergonic path until eventually we can break down that glucose into several steps and then finally get oxygen at the end as our electron acceptor. Okay, so we'll just remember that. This is not really like a major point that we need to cover now. We'll go through this a little bit more. But basically, when we get into cellular respiration, there are three major stages of cellular respiration that we want to cover. The first one is going to be glycolysis. Basically, every organism that does some form of cellular respiration does glycolysis. Even bacteria do glycolysis. Not everything will do the Krebs cycle, and not everything will do oxidative phosphorylation. But we'll talk about each one of these individually. The first one being glycolysis. So again, here's the pathway for cellular respiration. You can see that this bean-looking thing on the right side, that is supposed to be the mitochondria. And so anything that has a mitochondria, eukaryotes that contain mitochondria, can do some of these things that, for instance, bacteria can't. Right, so we have the Krebs cycle here, and then the electron transport chain. Those two things are part of like steps 2 and 3 from the previous slide. Glycolysis, if you observed, is actually occurring outside of the mitochondria. So you do not need to have a mitochondria if you want to do glycolysis. So again, bacteria or prokaryotes, they can do glycolysis, but they can't do the other steps. But we're going to talk about all of these steps because it's really important to understand how do eukaryotes generate just so much energy from consuming glucose or starch. So let's just jump into it and talk about glycolysis. We're going to focus just on this first step and see what glucose does in the beginning. So again, glycolysis, that's the starting point of the cellular respiration. Where does it happen? It occurs in the cytosol of the cell, outside of the mitochondria. It's really important to understand that glycolysis does not happen inside of the mitochondria. Glycolysis is the first starting point. If we break up that word glycolysis, you can kind of pronounce it weird, like glycolysis. That basically means that we are basically taking a sugar and we're cutting it in half, right? Lysis means to cut or to lyse. And so we're taking that glucose, which is a 6-carbon molecule. 6C means 6 carbon. And we're going to cut it up into 2 pyruvates. Okay, and pyruvate is a 3-carbon molecule. Okay, and so these are the steps that we're going to take a look at. I know this is really intimidating to look at first with all these like crazy names. You're not going to need to know the majority of these names going into like an AP exam. What you do need to know is what's the start and what's the end. And so this is kind of like a weird diagram because we start right here where my cursor is kind of floating around. Here's glucose, and then we kind of go around the right side and back over to the left side. So it's kind of like a weird diagram, but it's a really good diagram because it shows all this molecular structures, and then we end up with this thing called pyruvate. Okay, so just a review, we're taking glucose and we're splitting it into pyruvate. Let's talk about how that happens. In this diagram, we're going to focus on the carbons, and it's kind of hard to see, but the carbons are these gray molecules here. So you can see that this glucose has 1, 2, 3, 4, 5, 6 carbons, right? And the very first step, we have to take ATP to attach something to that glucose, and we actually used ATP in order to convert glucose into something else. And that kind of seems counterproductive because isn't the whole point of cellular respiration to make energy? Well, yes, but it's kind of like that thing where you need to overcome activation energy. So we have to like expend a little bit of energy to get more energy out. So we have to spend a little bit of energy in order to later get a payout from it. So we do have to invest ATP right here. And then glucose will undergo several changes where it'll still remain a 6-carbon molecule. And we've actually used 2 ATPs. You can see here's the other ATP right there in order to convert this glucose into fructose 1,6-biphosphate. Okay, so going from glucose all the way to here, this is still a 6-carbon molecule. We've had to use 2 ATP, so we're at negative 2 ATP. But at this point, what happens to this fructose 1,6-biphosphate, it basically gets cut in half, and then we end up with 2 3-carbon molecules. You can kind of see here, here's 1, 2, 3 carbons in this molecule and 1, 2, 3 carbons in this molecule over here. All right, so this molecule is now glyceraldehyde 3-phosphate, and this molecule will be converted into pyruvate eventually. But along those steps, there are crucial things that come out of this. The first thing is that we get something called NADH, and that kind of sounds familiar to NADPH, which we learned about in photosynthesis. And it's very similar. It's an electron carrier. It's basically like, you can imagine kind of like a truck just holding electrons ready to be delivered, kind of like an Amazon truck, and it's ready to deliver electrons to the last step of cellular respiration, and that's going to be the electron transport chain. So all you really need to know from the NADH is that it's going to be used later, but it's not going to be relevant right now. If I go back just a couple of slides, you can see here one of the byproducts of glycolysis, or not byproduct, but a product, is NADH, and that goes all the way to the end here to the electron transport chain. All right, so we got that going on, but then as we convert from this 3-carbon molecule, this glyceraldehyde 3-phosphate, all the way to pyruvate, which is still a 3-carbon molecule, you can see that we've actually generated 1 ATP here and another ATP here. So we've made up the ATP that we lost. But here's the thing to remember. We took that 6-carbon molecule and we cut it up into 2 3-carbon molecules. So this reaction actually happens twice. And since we cut this molecule in half, the second one will also do this step. So we actually end up generating 4 ATP. Okay, so we've generated 4 ATP, but we've had to invest 2. So the net payoff here is actually that we've got 2 ATP from this. Okay, so go back to the diagram here. You can actually see that one of the products is 2 ATP. Even though we had to invest 2, we got a payout of 4. So the total net profit here is 2 ATP. And then we also got 2 NADH because going through this one process, we generated 1 NADH, but this 6-carbon molecule, it got cut in half, and so we ran this whole reaction twice. So we ended up generating 2 NADH. Okay, so what's the final summarization from this? If you don't want to remember all of those steps, you got to at least memorize this last bit right here. If we take glucose, the major things that we get from it are 2 net ATP, 2 NADH, and then 2 water molecules that come off of it as byproducts. Okay, and then the final products are going to be 2 pyruvates. Okay, one thing to understand though is that sometimes you'll see in textbooks or in readings the term pyruvic acid, and that's basically just another term to pyruvate. So pyruvate is also commonly referred to as pyruvic acid. They're the same thing. So glycolysis, just as a review, is a 2-stage part. There's the energy investment part that we saw in the beginning, where we have to actually use ATP to phosphorylate compounds of glucose. And then the second part was the energy payoff stage, and this is where energy is produced by substrate level phosphorylation. That's that whole step that we saw where we get the ATP and NADH from that one glucose molecule. So that's this second part. The second part on the bottom, that's the energy payoff phase, and then the part up here is the energy investment phase. Okay. So here's our summary chart. We're going to be updating this chart with all the different steps of the cellular respiration. But for the very first part, we've got for cellular respiration and glycolysis, the input was 1 glucose, 2 pyruvate, 2 net ATP, and then 2 NADH. I would encourage you to create a chart like this in your notes just as a great study tool. Perhaps maybe even on your index card on your test when you take that in a week and a half or so. You could have something like this just to remember what are the inputs and what are the outputs of the cellular respiration. So we're going to keep updating this as we go through this PowerPoint. So the next part of cellular respiration is going to be this step right here. So we've taken those pyruvic acids or those pyruvates, and then we're going to convert them into something new so that we can actually use that something new in the Krebs cycle. So that thing is called acetyl-CoA. And acetyl-CoA, again, is just the first step of the Krebs cycle. But in that process, we're also going to make, if you observed, 2 more NADH. And those NADH will end up going here and that'll end up going to the electron transport chain. So just keep that on the back burner that we're also making NADH on the side. All right, so here's the first step of pyruvate oxidation. That's this next step. If oxygen is present, the pyruvate enters the mitochondria, the eukaryotic cell. So that's something really important to understand. We will talk more about this later, but in order for this step to happen, oxygen has to be present inside of the cell. So we have to have oxygen available. So that's why we breathe, essentially, is to get the oxygen. And the oxygen that we breathe ends up going right here into this step, into the pyruvate. If we don't have oxygen, acetyl-CoA cannot be made, and then we do not go into the Krebs cycle. So pyruvate is oxidized into something called acetyl-CoA, and acetyl-CoA is used to make citrate, or citric acid as well, in the Krebs cycle. And that's why sometimes the Krebs cycle sometimes is called the citric acid cycle, because we're making citric acid or citrate. It's the same thing. So what's the final end of the pyruvate oxidation, this step that we're looking at? Well, we get 2 carbon dioxides. That's a byproduct that we don't need. But we also get 2 NADH, and those are produced, and those are going to actually be used again later in the electron transport chain. So let's take a look at the next step. This is basically an oversimplification. We have pyruvate. We get 2 NADH and 2 carbon dioxides. Why do we get 2 and not just 1 like it says here? You have to remember that we ended up with 2 pyruvates after glycolysis, not just 1. So we do this whole step that we see here 2 times. So we get 2 NADH, 2 CO2s, and then we get 2 acetyl-CoAs at the end. And so this molecule is going to be the one that is going to then take the next step into the Krebs cycle. So let's go ahead and update our chart here. After pyruvate oxidation, we took the pyruvate, that was our input, and the output that we get is 2 acetyl-CoA, which is going to be used up. We're not going to really care about that right now. 2 CO2, which we certainly don't care about. This is a byproduct that we need to get rid of. And then 2 NADH, which are these electron carriers that we're going to use later. All right, next step we're going to focus in on is, okay, we're moving on. We've got this acetyl-CoA. That's just like this intermediate step. And then we're going to jump right into the Krebs cycle. And this is only going to happen if oxygen is present and we were able to generate acetyl-CoA. So let's jump into the Krebs cycle right now and see how that's going to work. The Krebs cycle is also called the citric acid cycle, like I've mentioned that before. Some textbooks call it only the citric acid cycle. What I'm seeing more now is that more resources are converting to the citric acid cycle, but for a really long time it was called the Krebs cycle. When I was in high school, so many years ago, it was also called the Krebs cycle. This occurs inside of the mitochondrial matrix, so on the very, very inside of the mitochondria. And then it turns acetyl-CoA into something called citrate. Okay, but citrate, if you have to remember, is also called citric acid. This whole process, we'll get to that later. It releases CO2, ATP is synthesized, and electrons are transferred to NADH and FADH2. These are products that we are going to use later. CO2 is a byproduct. We don't need it. We do get some energy out of this, so that's a good thing. We get some energy out of this whole Krebs cycle, and NADH and FADH2 are going to be used later. So this is all of the Krebs cycle, the citric acid cycle, and it's kind of complicated, as you can see, and this is like, I kind of included a picture of Einstein. This is the big brain diagram of it. There is a simpler diagram that I kind of simplified here. You can kind of take a look at this one later. If you'd like. This is more of an oversimplification. If you commit this to memory, that would be terrific, just because this is something that will give you all the intermediate steps that are necessary and all the products that are formed from the intermediate steps. But we are going to take a look at this one right here to really talk about the details of the Krebs cycle and how each and every single product is made. We've already talked about this step up here about how pyruvate gets converted into acetyl-CoA, but we're going to move on, and I have a little diagram that kind of shows you each step-by-step process of what's going on here. So the very first step that we're going to see is that acetyl-CoA is going to combine with a certain thing called oxaloacetate, and this is a 4-carbon acceptor molecule. This was regenerated from the previous cycle of the Krebs cycle, so this came in from the previous cycle, but it's recycled every single time we do this Krebs cycle. So just remember this is going to be the starting step, but also the final step is making this oxaloacetate. And each one of these purple circles represents a carbon. So just remember that we want to count the carbons. 1, 2, 3, 4 carbons here, 1, 2 carbons here. And so you can expect that what's going to be the product of this, we are actually going to make something called citrate, which is a 6-carbon molecule formed from acetyl-CoA plus oxaloacetate. Then we take our citrate, which looks like this. One note that I want to make here is that citrate, again, is commonly referred to as citric acid. Just like how pyruvate is called pyruvic acid, citrate is also called citric acid. Both are the same thing. So citrate will then turn into a couple of intermediates. So we get cis-aconitate and then D-isocitrate. You don't really need to know any of these. But in this process, we lost a carbon molecule. One carbon is lost as carbon dioxide, and because it had to combine with oxygen. But in that step, we were able to get another NADH, which is good because we want as many of these as we can get. So we lost the carbon, so now we're down to 5 carbons, 1, 2, 3, 4, 5, and this thing is called alpha ketoglutarate. Alpha ketoglutarate will then make another NADH, so in that process, we're losing carbon dioxide. At this point, you might see a little trend here, like every single time we're making an NADH, we're losing a carbon. So we've made another NADH here, we lost a carbon. So every single time we're making NADH, we've lost a carbon, and now we have a 4-carbon molecule. And that 4-carbon molecule is called succinyl-CoA, and that will then move on to form something called GTP. Now, GTP is something that we haven't talked about a lot. It's very similar to ATP in the fact that it is a nucleotide with 3 phosphates, but instead of adenine, it's guanine, right? So it's an energy-carrying molecule equivalent to ATP. We don't see it very often, but it is used, and it's pretty rare to see. And the reason why we don't see it is because every single time we do the Krebs cycle, it's basically, we only make like 1 or 2 of these, but we end up making like 36, up to 36 ATPs versus just like the 1 or 2 of GTPs that we make. So we have to keep that in mind. So that 4-carbon molecule is going to go through a couple of steps. This last step is basically, we've got everything that we want to get from this 4-carbon molecule. We are able to get another couple of electron carriers. This is another electron carrier called FADH2. And FADH2 is very similar to NADH in that basically it's carrying electrons for the electron transport chain at the very end. And then we are able to get another NADH. So we're reducing this thing the whole time. And when we're reducing it, we're taking something else off of this molecule. And so here are the intermediates if you want to see what's going on chemically. We have fumarate and malate being manipulated here. And then finally, malate is converted into oxaloacetate. And oxaloacetate is going to be the last step of the Krebs cycle. But it's also, again, the first step of the Krebs cycle. So this is how it's regenerative. This whole part going from the GTP up to oxaloacetate, that's kind of like this generative process, regenerative. All right, so if that was a lot, which I can agree with you that it was a lot of steps, you probably have to watch that a couple of times just to make sure you get it down. This is the summarized pathway per glucose molecule. This is something that I would expect at least everyone to understand and know this, that the inputs for the Krebs cycle were 2 acetyl-CoA, and here were the outputs. You got 2 ATP, 6 NADH, 4 carbon dioxides, and 2 FADH2. ATP is definitely useful. It's like immediately right off the bat, we can use that as an energy source somewhere. NADH and FADH2 will be useful later, but not at the moment. And then the carbon dioxide, that's not useful at all. We need to get rid of that through our lungs. And so we're going to update our little chart here. We've got the citric acid cycle, aka the Krebs cycle. The inputs, again, 2 acetyl-CoA, and then the outputs, 4 CO2, 2 ATP, 6 NADH, 2 FADH2. Okay, so that's what we have so far. And now we're going to jump into the very last step of the of cellular respiration, and that's going to be the electron transport chain. Okay, this is also called ATP synthase action. So this is where we're going to see a lot of ATP synthesis. If you've looked at this picture, we only generated 2 ATP here in glycolysis, 2 ATP here from the Krebs cycle. So we've only made 4 ATP, which is not a lot. But the electron transport chain, we're about to make 34 ATP. We say about 34 because we actually don't know how many we make. Sometimes we make 30, sometimes we make 32 or 34. So it's about that number, and it's not consistent. So somewhere around that range, that's how many ATPs we're going to generate from that. And so you can see all these electron carriers like the NADH that we made from glycolysis, the NADH that we made from pyruvate oxidation, the NADH and the FADH2 from the Krebs cycle. That's all funneling over here to the electron transport chain. So now it's the time to kind of focus in on this step and understand where is the bulk of our energy coming from. Like, so all of what we've been talking about has been leading up to this moment right here. All these steps from going from glucose, making all of these different products, we're going into the electron transport chain. So the formal name of what we're going to be looking at now is called oxidative phosphorylation. And oxidative phosphorylation includes 2 major steps. The first one is called the electron transport chain, and the second one is chemiosmosis. And chemiosmosis is the movement of protons, similar like in osmosis, from a high concentration to a low concentration. And it involves one of my favorite enzymes ever, ATP synthase. This is really important on making energy, and this is basically the bulk of where we get our energy. It's from ATP synthase. So this whole diagram, I know it's labeled here as the electron transport chain, but it does show on the right side with this blue enzyme. This is also part of the chemiosmosis. So this entire thing is the oxidative phosphorylation. And so there's a lot of key important integral and peripheral proteins within the membrane of the mitochondria. So this membrane that you see here, this double membrane, that's the inner membrane of the mitochondria. So this space above it is the intermembrane space. So you can imagine there's like another membrane out here. That's the outer membrane. It's not drawn up here, but you can kind of imagine that it's there. And then the space over here is the mitochondrial matrix. This is the very inner part of the mitochondria. And so the electron transport chain, which is going to be the part that we're going to look at first, which includes these red peripheral proteins. The electron transport chain is located in the inner membrane of the mitochondria. It's a collection of proteins, and as the electrons fall, proteins alternate between reduced and oxidized states. And so basically the whole goal of that is to make sure that we are moving protons to a specific part of the mitochondria. So what do I refer to as protons? They're going to be these H+. These are these hydrogens that get plucked off. These hydrogens, they're going to come off of a water molecule, but I'll talk more about that later and more detailed, like how does this work. And then we're going to basically put them all onto one part of the matrix. Now, this whole part should look familiar. We talked about something like this in photosynthesis, where we had ATP synthase and we had a membrane. So inside of the chloroplast, we were also making some energy there using proton gradient, just like this. Another thing to mention about the electron transport chain is that the crystate increase the surface area for the reactions. I've mentioned that before. This does not produce ATP directly, but it helps manage the release of energy by creating several small steps for the fall of electrons. Okay, so here's a little GIF. It's going to go a little fast, but basically you can see here that this is the inner membrane space of the matrix, and this is the mitochondrial matrix. And basically what's going to happen is that you see a lot of these protons kind of floating around in this inner membrane space. You also see the generation of ATP. If you focus here on the bottom right of the GIF, that ATP, here's ADP, and then ATP is going to be made. So the final electron acceptor of this whole process is going to be oxygen. So as the name implies, the electron transport chain, we are transporting an electron through a chain reaction of enzymes. So when we move this electron through these proteins, something has to accept it at the very end. And the final electron acceptor is going to be oxygen. Okay? So here are some more notes on the electron transport chain. One major function of the electron transport chain is to create a proton gradient across the membrane. I would say that's the most important function of the electron transport chain. The whole point is to make that proton gradient. As proteins shuttle electrons along the electron transport chain, they also pump protons into the intermembrane space. So those integral proteins, the red ones from the diagram before, they're pumping protons into the intermembrane space. And they are using the exergonic flow of electrons from NADH and FADH2. So where are all of these protons coming from? Well, they're coming from those electron carriers that we were seeing before, NADH and FADH2, which we've been building up this entire time. We've had a stockpile of NADH and FADH2. They are the source of the protons. And the proteins, part of this electron transport chain, are going to take those protons and pump them through onto one side of the membrane. That's going to create a gradient, and the gradient will then power the chemiosmosis and run through ATP synthase. Okay, so this is basically a diagram of how it's all going to work. Here's our little electron. Where did the electron come from? If you didn't see right there, there's NADH, and the electron is going to be right here. It's going to go right here into the first protein of the electron transport chain. And now we have got this protein, and then it's going to be in an excited state, and we've actually were able to move a proton using the energy from that electron across the membrane here. Then the electron gets kind of transported through all of these proteins, and every single time we move that electron, a proton gets pumped through the membrane onto this side. So again, where's the source of all of these protons coming from? We've got NADH here, we've got FADH2 over here, and they're all gathering these free protons and pumping them onto the other side. Then here's some more of these proteins pumping the protons across. And the electron ends up attaching itself to the hydrogen plus the oxygen to make water. So we're actually making water here. Water is kind of a byproduct here. It's not going to be really relevant for the rest of the cellular respiration. But with this proton gradient, now we're going to move on to this last step. This is going to be this molecule here called ATP synthase. So this next part, which is called chemiosmosis, ATP synthase, the enzyme that makes ATP from ADP plus a proton, it uses energy from the proton gradient across the membrane. So as these protons pump through here, every single time a proton goes through, ADP plus a phosphate turns into ATP. So ATP synthase synthesizes ATP. So it's kind of like in the name, right? And so this is kind of like how it actually looks like. This is, of course, an animated view of like what it looks like. But what would happen is that a proton would come in through from the bottom side. So this is actually like flipped upside down from what we actually see. A proton would come in from the bottom side. And then as it goes through, it's able to make ATP just by spinning it. So like every single time a proton goes through, it's kind of like a carousel, and it's kind of like pushes this ATP synthase, and every single time it turns, it gives it the energy, the mechanical energy, to force a phosphate and an ADP together to make ATP. All right, the proton ions flow down their gradient through ATP synthase. ATP synthase acts like a rotor, and when the protons bind, the rotor spins, and that activates a catalytic site to turn ADP plus a proton into ATP, just like what I said before. And this step alone will produce 26 to 28 ATP per glucose. This number, plus the number that we were able to get from glycolysis and pyruvate oxidation, and all those ATPs that we got before, ends up being our net total of like 34 to 36 that we see. Okay, and so that's basically the last step of the electron transport chain. There's a great video here that I will link in the description. If you're watching this on YouTube, I'll put that in the description below. I'm not going to play it right now. Again, for like copyright infringement, I can't actually show this video. So I'll put it in the description. I encourage you to check this out. This is kind of like an overview of basically what we've been talking about. So to fill out our chart here, we've got oxidative phosphorylation. This is the last step. Here are our inputs. We took those 10 NADH. Where did we get those 10 NADH? Well, actually, they're from our outputs. We had 2 here, 2 here, and 6 here. So that's 10 right there. And then there are 2 FADH2s that we got from the citric acid cycle. Those are our inputs. And then what do we get? We got 26 to 28 ATP. And that's actually a very good number from this one glucose molecule. So we got one glucose, ended up turning into 26 to 28 ATP through a very long series of reactions. And so sometimes we get 32, sometimes we get 36, ATPs. Again, that number kind of fluctuates. It's not necessarily cemented in stone that you're going to get a certain number of ATP, but it's usually around this number. A maximum number that you could possibly get is 36. On the low end, you'll get 30 to 32. All right, so here's kind of a flow chart of everything that we've been talking about so far, starting with glycolysis. And then remember in the beginning, I was talking about after glycolysis, you have to have oxygen in order to go on to the next step. So this is with oxygen right here. And then we ended up doing all of these steps: pyruvate oxidation, citric acid cycle, oxidative phosphorylation, which is basically these two parts, the electron transport chain and chemiosmosis. And then we got this many ATP. Now, what we're going to discuss next time in next class, because this is way too much for one period, is what happens if you have no oxygen. So what's the big question here? Like, how do we generate energy if we don't have any oxygen? So that's the big question that we're going to answer next time. I would definitely encourage you to understand all of these steps, especially the steps in glycolysis, in order to understand what we're going to talk about next class period.